All right, welcome back. Hopefully this thing is streaming right now. Please go ahead and open up the project you have set up for this meetup. And if you don't have a project set up, again, that's okay. For this meetup in particular, having a project environment will be especially useful. And I'll tell you why when we get to that stage. So again, just as we always begin, let's use this, just type in this code here. So it's remotes : install _ GitHub. And then the name of the package that we're using. I'm just going to go ahead and do it again. So mine worked out pretty well. Oh, sorry. Actually, let's repeat that. We need to specify that force equals true. Sorry, I forgot this. And this will ensure that any packages that we made to any updates we made to the package will now be reflected on your machine. So, okay. So hopefully that's worked for everyone. And then we can just load the package. And another thing, maybe I haven't said this previously, the reason I sometimes run things in the console versus in an R Markdown document is usually code that I don't particularly care about preserving. I just put it in the console and then it just disappears, and that's okay. So if you want to launch the slides for this week, you can do launch slides. And if you don't want to, that's okay. And to launch the Markdown file for today, just do launch _ project. And then it's number 5, so this is the 5th meetup. And again, so there's a lot of code here, mostly written by Paul, to nicely assemble all of the packages we need for today. So just go ahead and click play for the setup chunk. So that's up here. And again, it's super important while this is going on to keep an eye on your console. Because if R asks you any questions, it's going to ask you the questions here. So in this case, again, it's asking me if I want to update all, some, or none. I, by default, update all. Hopefully there are no more errors. Sorry, not errors, messages. And again, it's asking me, do I want to install this? Again, I just blindly type in yes or all or whatever it asks me to do. And that's going on. And so that worked pretty well for me. But then I already have a lot of these packages installed. So keep an eye on it, and if there are any warnings or not warnings, if there are any errors, just raise your hand, let me know. To kind of keep an eye on that, and we can begin the meetup. So today we'll be talking about two general approaches to performing enrichment analysis. Now, in this case, the focus is on, the focus is on performing enrichment analysis using RNA-seq data, but this logic can apply to any list of genes you have. So if it's microarray data, or you can even apply this to proteomics, SNP data, things like that. All right, so this is run. And I'm assuming everything's okay for everyone. And if it's not, again, you can just watch this, the walkthrough, and you can follow it along with it later on as you go through the code yourself. Okay, so one thing again that I did last week was if you want to annotate this R Markdown file, which I 100% recommend you do. You should first remember to save as in your project directory. So that will be for me here.
Is it okay to save it while it's still up, like loading stuff?
Yes. Yeah, that's a good question. So the question was, is it okay to save the R Markdown file while the code is still executing? And yeah, that's perfectly fine. So, and I know that I saved it to the correct directory because I can now see it appear here. So this is just a minor point to do with the package itself. I wouldn't worry about this in general, but if you want to have a repository of your own versions of these different project code files, then I recommend doing this. If you don't do it, then the next time you install the package, it'll all disappear. All right, so this next load and run thing here, this is covering a lot of what we already covered in the previous two meetups. And I'm going to run it. So what this does is it loads the RNA-seq dataset from one of the packages that's associated with one of the books we're following. And then it, wow, I did that really quickly. And then it performs this DESeq2 differential expression analysis to produce this object here, this res object, and that's the object that contains the results of our differential expression analysis. And just to remind you, this was an experiment where they knocked down some gene. I don't know what it was, but they knocked down something, and they compare the knockdown samples, the 3 knockdown replicates, with the 3 mock replicates. Okay, so I won't look at that object now because, or actually, why not? So if we just type it into the console, you can see that this is the output of all of that DESeq2 analysis. You can see log fold change, p-values, adjusted p-values, and the ensemble IDs for the genes. So that's nice. Last week, we went over different ways of performing data visualization using a lot of functions that are within the DESeq2 package. One really important diagnostic plot that we didn't cover is this thing called an MA plot. So I'll first show you what it is. So this is kind of strange. For whatever reason, it's just easier to copy paste this into your console and run it. And you can see it appear here. So this MA plot, so the MA stands for minus over average. And the reason for that is the y-axis is represents the log fold change, and division in log space is subtraction, so that hence the minus. And on the x-axis, you see the mean of the normalized counts. Each dot here represents a specific gene. And the blue dots are the dots with a significant adjusted p-value. One thing to take away from this plot, one of the main reasons this specific plot is used is it illustrates that when you look at the genes that have low normalized counts, you see that there's a lot of noise, meaning that like it's fanned out for lower counts. But as you get to higher counts, they all tend to, like there's less of this up-down noise. And the reason for that is when you do ratios of counts, if the counts are low, you're much more likely to get either very high or very low log fold changes. So it sounds kind of silly, but these estimates of log fold changes, these kind of naive estimates the DESeq2 gives us, aren't particularly reliable when it comes to low normalized counts. So we need to, it's kind of like when you do Western blot analysis and you get really excited that you have a 10-fold change. But if you look at the actual raw data, the bands are super light, super faint. So do you really trust those data? Or if you do lots of qPCR, and again, you get like a 10-fold change, but the CTs are in the 30s. Do you really trust that data? So it's kind of like saying, oh, wow, I saw a log 2 fold change of 4 here. But the normalized counts were just a handful. So do I really trust this? Now, there are a couple of strategies to employ here. One, you can just apply an arbitrary filter threshold and just get rid of these low normalized counts. The disadvantage with that is you're getting rid of information. So another, question. Does it depend on, yes, because the greater, the number of counts can be reflective of actual expression, but it can also be reflective of sequencing depth. So that's a good question. So there are a few things that influence the mean normalized count value. But either way, if the outcome, if what you end up with are low counts, then basically the lower the counts, the less you trust the data.
And is there a biological effect too where genes that have much higher copy numbers of the RNA change less would be subject to less regulation than
Um,
locally expressed genes?
I think so. Actually,
Like on that plot,
yeah.
the high counts, they're not changing much. Is it just a technical thing, or is there also biological or highly expressed genes still changing others?
So the question is, do highly abundant genes not change too much? I'm not really sure if I know what the right answer is. One thing I would speculate is a lot of high count genes would be housekeeping genes. Kind of makes sense. So you wouldn't expect those to be influenced by your intervention, or not as much as the non-housekeeping genes. So that certainly, I think, would represent these gray dots high up. The blue dots, they're supposed to represent bona fide biological effects. It doesn't look like much. Do remember that this is a log-fold scale, so it's still quite a bit. It's just that the estimates are a lot tighter. And I'll explain why, or I'll do my best to explain why there's this kind of, you can almost see, it's almost like an attraction towards a value of 0, this horizontal line here. Okay, so I mentioned that one strategy is to just get rid of these low count data. Another approach is to perform what's known as shrinkage, so shrinking the log fold changes. And I'll try to explain what that means after actually doing it. So before we go ahead and do the shrinkage, so I'll show you the steps now. Line 90 here has this function called results names, and that just gives you this really simple output here. It tells you what coefficients are in your model. So in this case, we just have 2 coefficients, and this is something we went over a couple of weeks ago. So the first coefficient, this is not linear regression, but if you want to think of it as linear regression because it's easier, you can. So in this case, we're just doing a simple comparison of 2 groups. So we'll have 2 coefficients. We have our intercept, and then we have our beta naught coefficient and our beta 1 coefficient. Now, the reason I'm showing this to you again is this coefficient name tells you that DE-seq2 is doing the comparison of mock versus knockdown. And this is another thing we went over a couple of weeks ago. It'd be better if it did knockdown versus mock. That makes more sense. The reason it's doing mock versus knockdown is because K comes before M. It's kind of silly, but that's just how R works. So one way to fix it, so actually that's what we kind of did previously here when we made this res object. We fixed it on the fly by adjusting the contrast so that instead of mock versus knockdown, it was now knockdown versus mock. In this case, because we, will have to recalculate this res if we want to use this plot MA function again. So meaning that we do the shrinkage on, we do the shrinkage on the previous object, the DDS object. So we need to change the order in DDS first. So to do that, it's pretty straightforward. One of the elements that we use to construct this DDS object is the column data, right? If you remember from a couple of weeks ago, we had 2 pieces of data that we used to construct this object. Count data, which was our count matrix, and column data, which told us something about these different columns, including the condition, mock or knockdown. And if you recall, that condition column, in fact, actually, we can just run it in the console, and we see let me try another thing. I'm glad I did that. So I thought it was a factor, and we just had to reorder the factor. Actually, it's not. It's a character vector. So meaning it's just, it's a character vector that contains these 6 objects. If you don't specify yourself that this is a factor, DE-seq2 through its pipeline will automatically convert it to a factor. But the problem is, when it automatically converts it to a factor, it sets the baseline as knockdown and not mock. So here we have to, by hand, do two things on line 95. We need to specify that this is not just a character variable, it's a factor. So it has levels. And while we do that, so we can use this factor function. And while we do that, we can also specify the levels. So in that case, when you do levels equals C mock comma knockdown, this specifies that mock is the kind of lower level and KD is the higher level. And to see what that means, you can run it. And then if you do call data $condition, you see now this looks a little bit different. You see, one, you have the elements of this variable, but then you also have this levels information. And it tells you that mock is the lower level and knockdown is the higher level. So now you have things in the right order. So that's great. So now we can just go through the same process again, and I'm going to overwrite the previous DDS object. You first make the DDS object using your count data, your new column data, and then you specify your design formula. You run deseek2, so it's doing all of these multiple steps. And once you've done all of that, you can confirm to yourself that the coefficient of interest, this beta1 coefficient, is now in the right direction. Now instead of comparing mock to knockdown, it's comparing knockdown to mock. So sorry for the slight, this might be a little bit confusing, but these kind of issues are very real in programming in general. So definitely it's great that deseek2 has these functions, or these functions might even just be from summarized experiment. It's great that there are these functions that allow you to do kind of gut checks along the way. So now we know we're comparing things in the right way. So we've done that to our DDS object. Now instead of, what did we do previously? Instead of using this results function on the DDS object, we'll use a new function called LFC shrink. And this performs this shrinkage. Now in this case, you specify the object name, DDS. You specify the coefficient you want to shrink. So that's our log fold changes. So that's another reason you do results names. You need to know the name of the coefficient. You can also specify the number. You can say it's coefficient 2. But when someone is reading your code and they see coefficient 2, that's a lot less informative than condition KD versus mock. So I prefer putting the name of the coefficient. And then this type here, I won't spend really any time talking about this, but this is the specific way they perform this shrinkage. And there's a whole paper dedicated to this, APEGLM. So I'm going to go through that so that you get this new object. It's taking a few seconds. Oh yeah, and it even tells you what that paper is. And I'll first show you the effect of this shrinkage. So let's go ahead and plot the first res object just to remind you of what that plot looks like. So that looks like this. We've already seen it. Now when we plot the shrunken log fold changes, we get this. This is a good thing. So it looks like there's a much wider spread, but then the y-axis scale has changed. So we can go toggle back and forth. So now all of these like log 2 fold change of 4 and negative 4, you can see those like low confidence high log fold changes have now been pushed towards 0. And this approach is called an empirical Bayes approach. And I've put a really nice reference here on line 112. That explains empirical Bayes using baseball. And the idea is, I won't give away too much because it's explained much more nicely in this write-up. The basic idea is you have, for example, batting averages for different baseball players. And let's say a certain baseball player has only played 3 games and they have a ridiculously high batting average. Is that estimate of their batting average, is that an accurate estimate of their ability? Probably not. Because we know, like let's say apparently the highest average you can have is 1. Let's say they have an average of 1 after 3 games. Would you call them the greatest player of all time? Probably not, because they've only played 3 games. But how do you, you instinctively know that or intuitively know that because you have a sense of what the average batting average is across Major League Baseball or across history. So you can use this empirical Bayes approach to arrive at a more accurate estimate by using those two pieces of information. This kind of prior knowledge of baseball averages and the actual measured average of this one player. And a very similar strategy is applied here, where you construct this prior expectation or prior distribution using log fold change information from all the genes in your experiment, plus the initial log fold change estimate for these genes, and that gives you this new revised estimate. And the way this empirical Bayes approach works for DE-seq2 is the genes with fewer counts experience more of a change in their estimated value. And it's kind of like, to use the baseball example, if someone just has played 3 games and you apply this empirical Bayes approach, you'll have a much bigger shift in the estimate of that person's batting average. But if they've played 10,000 games and you apply this empirical Bayes approach, the estimate won't change that much. That's because you're confident that if someone has played 10,000 games, their batting average at the end of those 10,000 games is a pretty good reflection of their skill. So it's kind of like this. So you have all the experienced batters up here and the inexperienced ones here, and these are the ones that experience the greater change in value. And this empirical Bayes approach is used across omics data. So it's, I know there are people here who are familiar with Limma. Limma uses this empirical Bayes approach to calculate its what are known as moderated t-statistics. And it's the same overall idea of borrowing information from the data set in general to arrive at more accurate or believable estimates. Anyway, like I said, this Variance Explained blog post on baseball does an amazing job of describing all of this. And the final thing with this kind of this step is to convert this res shrunk object to a data frame. Also, I should say that an earlier version of DE-seq2 used to automatically do this shrinking for you. Now, they don't. And the reason is there have been developments in the specific algorithms used to do this shrinking. So they've kind of made it a little bit more difficult for you, but that's to reflect the development since the publication of that original paper. And they even say, if you go through the vignette, even if you don't take anything away from like what the shrinking means, the DE-seq2 people recommend doing it for 2 reasons. One, for data visualization. So if you want to visualize your data, do the shrinking. And for enrichment analysis, which is what we'll be doing. Okay, so I mentioned this enrichment analysis, and I said that there were 2 approaches that we'll be covering. One is, the first approach is known as overrepresentation analysis, and the second is gene set enrichment analysis. So overrepresentation analysis, the idea is pretty straightforward. You have these various collections of genes. So in this case, one database of these collections is the gene ontology, geneontology.org. And it has these various gene ontology terms, and each term consists of a list of genes that are all related in some way, some meaningful way. So either they're functionally related or, they're in the same pathway, or they're put in, they share some kind of relationship, and many such terms are constructed in this way. And there are many Go terms, but broadly, these Go terms fall into 3 broad domains. There's a molecular functions domain, so this gives you information on functional activities. biological processes, so larger biological roles. So for example, photosynthesis would be one biological process. And then the cellular component, like where in the cell are these proteins doing their job. And to kind of get a picture of these various Go terms, so for example, the hexose biosynthetic process. So these Go terms are arranged in this hierarchy. And the lower ones are the more specific, smaller Go terms, and as you go up, you can see you get more and more general Go terms until you arrive all the way at the top, not shown here, at basically 3 broad Go terms, which are the 3 major domains shown here. So all of these Go terms, you can use these to ask the question for, let's say, hexose biosynthetic process, that Go term, in your gene list of interest, which we will define in a minute, is there an overrepresentation of genes that belong to this Go term? Meaning, let's say you had a gene list of 1,000 genes of interest that passed some series of filters. And the expected value, the number of hexose biosynthetic process genes was 5, say. And you saw 7 genes. Is that an overrepresentation? Is that something to get excited about? So luckily, you can quantify the overrepresentation, or your surprise, by using one of various tests, but the hypergeometric test or its variants are very popular. And again, I'm going to skip the detail of what a hypergeometric test is because it's a very well-established test that has, and there are lots of great references that describe it, usually using colored balls in urns, but again, that kind of describe genes really well. And if you want to know more about that, you can email me and I can send you these references. But a simple Google search will give you really nice explanations. Basically, it's if you see this test quantifies how unlikely it is to see the number of genes that belong to a certain Go term in your query list, and that's it. I only showed you the Go database, but then there's KEGG, Reactome. There are different such databases. Go is, I think, the most popular one, so we'll use this. So we need to prepare our data to do this Go analysis. Sorry, this overrepresentation analysis. And so these lines here are very similar to what we did last week. So last week what we did was we used this Biomart package to derive gene symbols or gene names for ensemble IDs. Today we're going to use these ensemble IDs that are in our res shrunk data frame, use these ensemble IDs to get the corresponding entree IDs. And that's this here. Again, I'll skip the details of these lines because this was covered last week using this getBM function. So we use as a query, we use our ensemble IDs, and then we are retrieving ensemble ID, external gene name, which is the gene symbol, and the entree IDs. So I'll just run that here. And the reason we're doing that is because the database we'll be using predominantly uses entree IDs. So we need entree IDs for the subsequent code. And then the final step for this one was to do a left join. So let me just show you this new object, this ensemble to entree object, what that looks like. Oops. So that's just this object here. So you can see it has 3 columns, the ensemble gene ID, the external gene name, and the entree ID. So unfortunately, there are a lot of missing entree IDs, but we saw this last week when we looked at gene names. There are lots of missing gene names. So we can use this data frame, this ensemble to entree data frame, and our resdf data frame. We can perform what's known as a left join, and this is another thing we covered, I think, last week. And this left join combines these 2 data frames. So I'm just going to run that, and then if we look at resdf, we see that we have ensemble IDs, we have all the information that our DESeq2 produced. We have our external gene names, and now we have our entree IDs. And these IDs will be important to do the overrepresentation analysis. Okay. I think you've already seen the code, but anyway. So we saw that there are lots of missing terms in the entree ID column. It'd be nice to get rid of them. What's one way of doing that? No. Filter is one way. So that we can do filter, or there was a function that we brought up. So with filter, we can easily do it with filter, but we have to write a little bit more. There's one dedicated function to doing this in the tidyverse that we brought up. It has I would say memorable, but maybe not so memorable. It's called drop NA. So it's nicely named. So if you apply drop NA to your data frame in general, any row that has an NA in any column will be dropped. And that's a little aggressive. You might end up with just an empty data frame. Probably not. But you end up with a very small data frame, and also you don't know which rows were, necessarily why the rows were dropped. So you can specify within drop NA which rows you want to drop based on missing values in the named columns. So in this case, I want this data frame to contain no missing values in the entree ID column, and I also would like to have all the adjusted p-values be actual p-values, so no missing values there. So this is just a judgment call. I decided that I don't want NAs in these two columns, but if I see NAs anywhere else, I'm okay with that. And I use that to override this res_df object. So another question is, just because of the way these things are named, and it's not uncommon to have repetition in your data frame. So that's another thing, this line 152 here. This line of code is something that is really useful to do periodically through your analysis, just to make sure, just to see if there are any duplicates in your data frame. Because a lot of times there are lots of packages where if you put in duplicate information, the functions just won't work. They need to have unique information. So in this case, I'm not especially sure if the functions that we're going to use later on require unique, but it's I've put this here anyway, just to ensure that all the entree IDs you have in your data frame are unique. So one way to do that is to run this line of code here, and this tells you how many unique elements, how many unique entree IDs there are. So again, just like last week, if we work inside out to understand this, that can help. So if we do this resdf $entree ID, that's the innermost line, we just run that in the console. You can see it just gives me this list of entree IDs. Great. Now I can use that as input to this function called unique. And what unique does is this gives me a very similar output, except unique will collapse any duplicates or triplicates into just one. So it gives me all the unique entree IDs in this case. Now if I want to see how many of these unique elements there are, I can then use this as input to this length function. And if I run all of this, I'll just get one value. And this number here, 13711, tells me how many unique entree IDs are in my data frame. Now, if I want to compare that to the total number of entree IDs that are in my data frame, I can just do n rows, so the number of rows, because I know that there are no missing values for entree ID because I got rid of those in the previous line. So if I run that here, this n row, I now see that I have another number, and it's very similar to the first one, but it's not identical. So that means there are at least a few duplicates or maybe triplicates in my data frame, and I want to get rid of them. Now, getting rid of information is always a little bit tricky. How do I do that? Like, what strategy do I use? So in this case, I decided to, for entree IDs that are non-unique, I decided to keep the entree ID with the lower adjusted p-value. Kind of makes sense to me because it's the more significant one. So to do that, I took my ResDF data frame. You can arrange it in p-adjusted, so it'll be in ascending order of your adjusted p-values, so most significant to least significant. And then there's this other really nice function from the tidyverse called distinct. And distinct will it'll go through your data frame. And so in this case, I give it the entree gene ID column as an input. So it'll go through the entree gene ID column, and it'll pass by all unique values. It'll ignore them. The minute it comes across a duplicate or a triplicate, it'll drop it. So it'll get rid of it. So this is a nice way of ensuring that the output only contains unique values. Now there's this additional parameter here called .keepAll equals true. It's essential to specify this because if you don't, you will end up with a one-column data frame of just entree IDs. This is something I've done a lot and keep making this mistake. So if you run all of that and override your ResDF, you should now have a data frame that only contains unique values for your entree IDs. And to confirm that this is true, we can run the previous lines, 152 and 153. But in this case, instead of running them one after the other and then comparing the two numbers ourselves, we can just use this familiar equal to comparison operator. And that just asks, is the stuff on the left-hand side equal to the stuff on the right-hand side? And we know the left-hand side will be one number, the right-hand side will be another number. And so if we just run all of that, we should get true. And we do get true. So it's a lot of preparation of our data frame. So before I do the overrepresentation analysis, any questions? Okay. So I mentioned to do this overrepresentation analysis, you need a couple of things. You need a query set of genes. And this, you're asking the question, in this subset of genes, is there an overrepresentation of certain Go terms? So that's up to you. What is your subset of interest? So in this case, I decided that it was only going to be genes that have an adjusted p-value less than 0.05 and a log fold change greater than 1. Okay. And you can change this to, and I'm calling these significant genes, but you can adjust, you can filter your data frame in any way you like. And for the overrepresentation analysis, all I need is, once I do this filtering, I just need the names, these entree IDs. I don't need any other information. So to do that, there's another really nice tidyverse function called pull. And pull, as the name suggests, it just pulls all the values from the given named column. Right? So if I do that and save it as an object called siggenes, if I run siggenes in my console, I now see I don't have a data frame, I have a vector of integers. Okay? And now, doing the overrepresentation analysis is just one line, well, separated across, but really one line of code. And it's using this function called enrichGo. And enrichGo, I'm not going to scroll back up, but it's from this package called Cluster Profiler, which is one of the packages that you've installed today. So the way enrichGo works is it requires a few inputs. The first input is this parameter called gene. And gene is the subset of genes you're interested in, your query subset. So in this case, it's siggenes. It's important here and sadly I worked this out through trial and error, you need to convert it to a character. And this is one of those annoying things in programming in general. A lot of these functions in general require things in a certain data of a certain type or things in a certain format, and so you need to be accommodating. Or you need to adjust your input so that it matches their requirements. So they require these query genes to be characters. So we can use this function, base out function called as.character, to on the fly convert sig genes to character vector and not do what I just did. The second is they require a database that contains annotations for these for human genes and Go terms and other such things. And this database here, this org.his.eg.db, now that is also comes from a package that we installed today, and it's a comprehensive source of information about the human genome. There are similar such databases for other organisms. So for mouse, it would be org.mm.eg.db or ms, one of the two, and other such organisms. So we need to specify a database. Then we need to specify this thing called a universe, and this is our background set of genes. And in this case, our background is just simply all the genes that are all the entree IDs. So not just the entree IDs that we are querying, but all the entree IDs in our results table. So that's easy to get. You just do res_df $ entree gene _ ID, remembering to again convert them to a character vector. So we do as.character. Then we need to specify the ontology. So this is going back to those three big domains, Go domains. So this is molecular function. If you wanted to do cellular, it would be CC. Biological process would be BP. So I'm just doing MF here. That's the one I'm interested in. And then finally, there's this optional argument here called readable equals true. And this, if we don't set this to readable equals true, we get the output. Output table will have the entree IDs. If we do readable equals true, it'll convert the entree IDs to gene symbols. So it's just nice to see gene symbols, again, because we can recognize them as opposed to these entree IDs. All right, so if we run all of that, it takes a few seconds. And while it's taking a few seconds, are there any questions? Okay. We now have our results, this go_aura. And again, if we just wanted to see the first few terms, we can use this head function. You can see these are the Go terms that have come up as being significantly regulated in our query gene subset. So they're the Go term IDs, they're descriptions. Gene ratio, it tells you of the of your query number, I believe. It's the number of genes that are in this Go term. And then there's a p-value, adjusted p-value. So yeah, so that's really it. So those are the top Go terms that come up using this overrepresentation analysis. And I forgot to mention, but there are many approaches to doing overrepresentation analysis, many packages you can use. The reason I use this specific one, Cluster Profiler, really is because of its visualization tools. So we have this Go_aura table that's the result of the ORA analysis, overrepresentation analysis, but visualizing it is always nicer. So there's one function called dot-plot. And another good thing about Cluster Profiler is the functions that it has for visualization work really nicely with ggplot functions. So if you aren't happy, or if you want to change the elements of the output plot, you can use your familiar ggplot tools. So in this case, and it also works with this pipe. So I can do Go_aura, pipe it into this dot-plot function, and show category tells me how many of these Go terms do I want to see. So it's specifying a maximum of, in this case, I put 30. And then I can put a little title, dot-plot for ORA. So again, let me just run that in the console. Oops. And you get this dot-plot that looks like this. Let me zoom in. So you see this. It adjusts it. So you see the most significant terms up here. And as you go down, It color codes the adjusted p-value and the number of genes. So this is an attractive plot. There's another one that unfortunately doesn't work so well for these data, and that's using a heat plot that works very much like the dot plot. And what this shows you is, it shows you, and it doesn't work so well because there are so many query genes. On the x-axis are all your genes, and the y-axis in this case are only the top 5 Go terms. And these black lines show you membership of these genes. So you can see that in this case, whatever this gene is here that's hard to see, it's in these 3 Go terms. So this is a potentially useful visualization. An even better one is to use this upset plot. And that looks like this. Or actually, that gives you slightly different information. This one tells you how many genes there are in each term. Not just in each Go term, but it also tells you how many genes there are. For example, in this case, there are 30-something genes in these 3 different terms. So you can see, because these terms aren't necessarily exclusive, so there's overlap in gene membership. So this quantifies that overlap. So that's kind of interesting. So anyway, there are these tons of these built-in visualization tools in Cluster Profiler. All right, so that's overrepresentation analysis, and the last one is this gene set enrichment analysis. I'm just going to go ahead and go through that, and then if there are any questions, you can try to answer them at the end. So gene set enrichment analysis uses a different approach, obviously, to overrepresentation analysis. So we saw in overrepresentation analysis, we need to supply a query list of genes. We get rid of a lot of useful information, though. We're just giving it a list of genes, but then there's no information about fold change or p-values or anything like that. So gene set enrich, and another thing is we are arbitrarily supplying a list of genes or a subset of genes. Gene set enrichment analysis is different in 2 fundamental ways, or different in at least 2 fundamental ways. One, you use all your genes in this case in your experiment as input. And 2, this GSEA takes into account the rank of your genes. Now, you need to decide how you rank your genes. You can do it based on fold change, p-value, test statistics. So to just quickly go through, so the original approach was described in 2005 in this paper. And you can see that there are 2 comparisons, phenotype A, phenotype B. They've done some kind of microarray experiment. And these genes are ordered in some way. I'm going to say fold change. Now, another thing you need for gene set enrichment analysis is its so-called gene set. And this is kind of like a Go term, except these gene sets are defined by, I believe, the Broad Institute. And so a gene set could be, for example, you can think of this as oxidative phosphorylation. That could be a particular gene set. And these horizontal lines represent members of gene members of this gene set. And the location of the horizontal lines match the rank of that gene in this experiment. So let's say this was AKT. AKT belongs to gene set S. That means AKT was pretty high up here. And this was like mTOR, say. It's pretty low. So these lines, these horizontal lines, show you where the members of a given gene set, where they are in your ranked list. Okay? So when we go from this, the next thing is to, so just looking at this, if you saw, for example, a bunch of horizontal lines up here, that would indicate that the majority or many members of gene set S are crowded towards the top end of your ranking scheme. So if you were ranking your genes based on fold change, you then say, oh, many of the genes in gene set S are upregulated significantly, very highly in my experiment. So that means gene set S, if it represents, let's say, oxidative phosphorylation, I can then say, oh, maybe OXPHOS itself in general is induced. Conversely, if you see a clumping of these horizontal lines towards the end, you could then say the opposite. Oh, OXPHOS is significantly suppressed. A boring picture would be to see this random scatter of horizontal lines or an accumulation of horizontal lines in the middle, because the middle just means 0-fold change or around that. That's boring. So to quantify it, what they did was, what you can do is you can take this gene set with now these horizontal lines that correspond to the rank in your experiment, and you can turn it 90 degrees. You can see here, and so this, and what you do is you go through your gene list from the highest rank to the lowest ranked, left to right. And if you see a gene that belongs to gene set S, you add a little number here. And that number is proportional to the rank. So for example, if as I go across the first 3 or 4 genes, there's no change in my score. It's still 0 because I don't see anything. And then I see my first gene that belongs to gene set S. And it's a pretty high rank. It's up here. So I'll add a proportionally high number. Oh, and then I see 2 or 3 more. Great. So I'll continue to add my number. But then there's a stretch of no gene set S genes. So then if you don't see any gene set S genes, you subtract. So then I go down. But then there's a big cluster of gene set S genes here. So I keep adding. And then it's just kind of like sparse territory with a few genes, but they're all low ranked. So I go down. And so that way you get this kind of distribution. And from this, you can calculate what's known as an enrichment score. And the enrichment score is just the maximum height of this plot. So the higher the plot, the more significantly regulated a given gene set is. And you can also see in this case, the enrichment score has a positive value. But if everything were clumped here, you would see kind of the mirror image of this. This would be below the x-axis. And then you'd have a negative enrichment score. And that's it. That's how enrichment scores are calculated. All right. So to do this in the last 8 minutes, and the final thing is we have enrichment scores, but then one thing that influences these enrichment scores are the size of our gene sets, and obviously gene sets are of different sizes. So we can, there's a normalization that is performed to produce what's known as a normalized enrichment score that takes into account this difference in gene set sizes. So then you can compare one gene set to the other, results for one to another. All right, so to do this gene set enrichment analysis, we can use this package called GSEA. The F stands for fast. And to prepare that, so I'm taking my ResDF data frame, and now I've decided that I'm going to rank my genes based on log fold change. And again, to go all the way back to our log fold shrinkage, you can see why hopefully the shrinkage helped here. Because if we hadn't done the shrinkage, a lot of the high ranked genes would have been, the ones with the really high fold changes, would have been genes with really low read counts. So the ones we're not too confident about. So they would have really influenced our gene set enrichment analysis here. But by performing the shrinkage, those high fold change, low count genes now become low fold change genes. So then they go from being ranked really highly to being ranked much lower. So their influence on our gene set enrichment analysis is muted. All right, so we arrange based on log fold change to go from highest to lowest, so most positive to most negative. And then in this case, we just want to select the entree gene ID and the log fold change. So we have a 2-column data frame called 4_GSEA. Now, we need to supply to this FGSEA function ranks. What are ranks? Ranks are basically just a vector of these log fold changes that have names. Now, that was a really bad description. Let me show you ranks and then try to show you what that means. So if we just look at 4_GSEA, we have this. We have a 2-column data frame, right, with our entree gene IDs and our log fold change. We can use this function called dframe, and I'll just use it to make this ranks object. And now if we look at ranks, we see that it looks it's kind of taken our 2-column data frame and made this vector of log fold changes, but each log fold change has a name, and that name is the entree ID. That's it. And that's how we have to supply this. We have to, again, we have to supply data in the format of things in the format that are required by the functions. There's this thing here. So I mentioned gene set S in this case. What does that mean? So these are all curated sets that the Broad Institute has curated. And they put them into various collections. And there's a dizzying number of collections. So where do you start? One useful place to start is, that's recommended, I believe, by the Broad, is to start with a hallmark gene set. And this, I think, consists of like 50 gene sets that cover a broad spectrum of biology. And then based on the results of that, you can then dig deeper into other gene sets. So one approach is to just download the set of gene sets, so the hallmark set. And I've put this URL here that takes you to this collection. And the collections come in either, it's a collection of gene symbols or entree IDs. So you just have to make sure that you get the right one. So this H stands for hallmark. It's the most recent version because these things keep getting updated. And then it's entree. And then we can use this function called GMT pathways to get the pathways. Hopefully this works. So, I mentioned why do we do things in projects. So I've already downloaded this and put it into a new folder called data. And if you don't know how to make a folder in RStudio, it's easy. You just click on new folder and you give it a name. Now, because I'm in a project environment, the path that I put in is just the relative path, meaning I just put data/name of the file. In this case, there's only one in data. Now, if I weren't in a project environment, depending on where I was and what my working directory was, this path here might end up being super long. And I'd have to keep track of it. But here, since I'm in my safe project environment, the path is just data/name of the file in data. All right, so then I save that all as pathways.hallmark, and it's this crazy long object here. So you can see it's a list containing 50 gene sets. So we can see the last member of this list is pancreas beta cells. And you can see all the genes that are in this gene set. And if you scroll up, you'll see other gene sets with other genes. OK, running FGSEA is now super easy. You just do FGSEA. You give it the pathways. So in this case, so you set pathways equals to this list of 50 pathways, 50 gene sets. And then you specify what you are using, like your input genes with their rank metric. So the input genes are the entree IDs and the rank metric is a log fold change. And both these pieces of information are in this ranks object we just made. And finally, there's this nperm term that I didn't cover. So in this case, we calculate an enrichment score, and that's great. But what does an enrichment score by itself mean? Is it good? Is it high? Is it low? So one way to do this is to take this gene set S, work out how many members are in this gene set, and then just randomly permute other gene sets that contain the same number of genes but different gene members. Right? Just randomly put in whatever genes into these genes. So then you have, so let's say gene set S has 50 members, you construct another 1,000 collections of 50 genes each, just randomly chosen, and then you repeat this whole process to calculate an enrichment score. And so then you construct a null distribution of enrichment scores based on these randomly selected genes. And then you see and that distribution will have an expected or an average enrichment score, and then you compare that to the enrichment score for your for this given gene set and your genes, and you ask the question, how unlikely is it to see this number? And the more unlikely it is, the more the lower the p-value. And so in this case, you can specify how many how many numbers your null distribution contains. So in this case, if I do nperm, it's 1,000. It'll be a distribution containing 1,000 enrichment scores, which seems to be a reasonable number to start off with. And as you can see, it did that. That's why it's called FGSEA, because it did it in like less than a second. All right, so we have this FGSEA res object that I just made, and we can turn it into a data frame. So we can do FGSEA res, pipe it into as_tibble, and then we can arrange it in descending order of our normalized enrichment score. So I'm just going to quickly do that. And now if we look at the output, we get this. So we see these are the 50 members of the hallmark gene sets. P-value, adjusted p-value, enrichment score, normalized enrichment score, and other information. So you can see that the top term is epithelial mesenchymal transition. It has a positive enrichment score, so that means it's associated with genes that are upregulated by this knockdown, and the adjusted p-value is is really small. So it's believable that this is a term that is upregulated by this knockdown. And if you want to visualize it, you can, by converting it to a data frame, you can then use ggplot. I'm just going to, over time, so I'll just race through this bit. This is code that is pretty familiar to ggplot code you've seen before, but I'll just show you the output. So if you run that, you get something like this pretty plot here that shows you visually what the most interesting pathways are, which ones are significant, which ones aren't significant. And kind of as, quote, homework, you can go through this code to make sure you understand what each line is doing. And finally, there's this function called plot enrichment within the FGSEA package that works like this. You give it as an input one of the members of the hallmark gene set in this case. So in this case, let's say I do the top term, hallmark epithelial mesenchymal transition, and then you give it the ranks. And if you run this, you get this, right? And this looks kind of like this one. And so you can see that there's this peak close to the high-ranking genes, so it shows that this pathway is upregulated. So that's an interesting upregulated pathway. I'll show you what the profile of an interesting downregulated pathway looks like. So that's this. It's kind of like the mirror image, right? And again, you can see there's clustering towards the low-ranked genes. And finally, I'll show you what an uninteresting pathway, something in the middle, looks like. It's kind of like this. Everything's sort of scattered. And that's that. Are there any questions? Yes?
So what actually is the program to run the GSEA? You can download it and keep it very easily. So what do you need?
That's a good question. So the question was, you can use a GUI interface to run GSEA, and you can even run it through the Broad website. I think that's a great question. I would say the reason I prefer doing it in R is it's more, there's a greater footprint and history of it. But it's just generally my answer would be, my preference for doing it in code versus a point-and-click interface is that this is a lot more reproducible. Because if I had to then, for example, tell a colleague how I did my analysis, I could either just send them the code, or I could describe what I did using that interface. So, and also just for me, I'm just challenged when it comes to these different tools. So rather than try to learn how to use these various tools, and they all require things of a certain format, it's just easier for me to use one mega tool, in this case R, and then, yeah. But that's a great question. I would say there's no strong reason to prefer one over the other.
But just in terms of the output, the results get exactly identical.
It should be. That's a good question. I haven't tested it, but it really, I hope it is identical. Any other questions? All right, thank you for your attention. Hopefully this got live streamed, and